Hi, my name is Diamond O'Donovan and this is my Gummy Bear Lab. So I put one green gummy bear into a solution of just 1/4 cup of water. And then I put another green gummy bear into a solution of 1/4 cup of water and as much salt as it needed until it dissolved. I poured it straight from the big container, so I'm not really sure how much salt ended up being in there. But my results were as expected. My gummy bear in the water solution is huge. Try to show it here. And very clear. It's also very fragile, as I found out a little bit earlier taking it out. And this is my salt water gummy. So it looks about the same size, maybe a little bit smaller, can't really tell. And I had both of these in for a little over 12 hours, just until I had time to make this presentation. So the results were as expected. This is due to osmosis, which is the movement of water molecules from an area of higher concentration to an area of lower concentration over a semi-permeable wall. The gummy bear is the semi-permeable wall here, so water is able to flow into the gummy bear. So as stated in the lab description, gummy bears are made up of sugar, starch, and gelatin. Since there is no water in the gummy bear, when it was put in the cup of water, the solution was hypotonic compared to the gummy bear. That means there was a greater concentration of water molecules in the solution compared to the gummy bear. Water flowed into the gummy bear to create an isotonic solution. So when there's an area of higher concentration to lower concentration, the objects want to create an equilibrium. So water flowed into the gummy bear to create an isotonic solution, and it eventually created that, as seen by the size of the gummy bear. You can also see there is like a change in color. It was almost clear with just like a tint of green compared to the dark green that it had before, which kind of explains like that there is more water in there due to the color change. In the salt water solution, the gummy bear, I couldn't tell if it stayed exactly the same, possibly shrunk, and I think because everyone has like a little bit of a different salt water solution, just due to however much dissolves, because temperature and the amount of stirring plays a part in that. So if you stirred more or faster, longer, you're going to get more to dissolve in there than whoever just kind of slowly does it, just because the salt will eventually just sink to the bottom. The gummy bear could have been already in an equilibrium state, which means that there is the same amount of water molecules in the gummy bear and in the saltwater solution. So that's why I couldn't really tell if the gummy bear got any smaller or bigger. It looks almost exactly the same to me. But I was surprised by how much the big gummy bear, or the water gummy bear, grew. I wasn't expecting it to be as large as it is. Obviously mine was in there a little over 12 hours, so I'm sure it just kept growing. But it eventually stopped, and it's now so fragile. Like, I don't think that the wall of the gummy bear can even hold as much water that even like came in there. As soon as I picked it up, I kind of broke it all over the place. So in conclusion, the gummy bears were originally the same color, the same size. They were a little bit stiff and bright green to begin with. And then afterwards, because of osmosis and wanting to create an equilibrium of the two objects, the gummy bear that was placed in the water solution grew because water moved into the gummy bear to try to create an equal amount of water particles. And then the gummy bear in the saltwater solution probably stayed about the same size because the saltwater solution didn't have as much water and had as much particles in the gummy bear and in the saltwater solution, so it stayed about the same size. So my experiment came out as expected, and it's because of osmosis.